Here we are, Eternacon 8. It's so great to see, on the one hand, so many old friends who we've seen. I've met in prior Eternacons going back, wow, 7 or I guess 8 years now. And I also want to welcome many new friends who are coming here for the first time. I see some new names and faces, so you're extraordinarily welcome to come join this community. So Eternacon, for me, as a scientist, this is my favorite scientific conference every year. It's such an unusual event. It's a hybrid between a gaming convention and a scientific conference. But in any of those dimensions, it's the most exciting for me. And there's an additional dimension that I've come to value more and more, which is the community that we have. And I think you'll see that several of the sessions this year, but also I guess in every Eternacon going back 8 years, there's been this real emphasis on finally getting to interact with people who we talk to online and interact with in terms of science, but also in terms of solving puzzles. It's a really special thing to be able to see everybody all at once in one event. So I guess in these opening remarks, I wanted to reflect a bit on what Eternacon has meant for Eterna. Okay, so A, of course, it's this great gathering of the community, but B, it's also been, like, I would say that it's become the key event in which core ideas happen, initiatives are brainstormed that then have an impact 2 or 3 years later. They really blossom in a later Eternacon. And so I started thinking back to what's happened, going all the way back to 2019 Eternacon, which was the last Eternacon where there was a in-person part of it. It was a hybrid event. Several of us met at Stanford. Several of you also came in virtually. And I just wanted to note a few examples of how ideas that I think I had never heard of then, or many of us had never heard of until then, came into play and then now have just blossomed in the last few months. So as one example, I guess for me as a physicist who jumped into biology about 20 years ago, I've always been amazed by a molecular machine called the ribosome, which is an RNA-based machine that makes proteins in all living cells. And it's probably the most ancient molecular machine that we know about, and it's potentially the most sophisticated molecular machine that any biologist knows about. And when I looked at it, and when others have looked at it who've come into biology, they say, wow, to really understand how that thing works, I'd love to be able to design it, like maybe even make it better. Like, how amazing would that be that we could surpass 4 billion years of evolution by nature if we could make it better, more stable, produce alternative proteins. That would really demonstrate that we understand biology, and maybe would have even practical consequences by allowing the ribosome to make new kinds of materials. And in 2019, we invited several really the world experts in ribosome research to come and join the meeting. I think that was really the first time. Folks from Northwestern came. They had a huge ribosome engineering effort. And they came in person and met Eterna players like Ome and others. And they started to initiate, and there was a big discussion of some pilot puzzles for some ribosome design puzzles that we had put up before in late 2018 and 2019. And that resulted in challenges that we're now calling the open ribosome challenges. And in the discovery of molecules that we're calling superfolder ribosomes. And the first paper ever, to my knowledge, showing that you can rationally redesign the ribosome to be more stable, to allow for better folded ribosomes that have enhanced function outside cells, and ribosomes that allow for bacteria to grow better, even when they're doused with alcohol. That paper has come out in the last year, and you're going to hear Andre Kruger talk about those pretty fascinating molecules tomorrow. In addition, Alana Shephardt, now at Berkeley, the leader of the Center for Genetically Encoded Materials, attended virtually Eternacon that year, along with Sarah Smaga, who's the director of that center. And they were inspired to set up their own puzzles, which have been launched this year. And you're going to hear about CGEM, their center, and several really, really cool puzzles from Sarah tomorrow. So there, that's, this wouldn't have happened if it wasn't for Eternacon, I would say 3 years ago. In the same Eternacon in 2019, that was the first time we talked about stabilizing mRNA molecules. And some of you might remember this. We talked about trying to get Eterna players to stabilize mRNA vaccines for a pandemic. This is in 2019. Of course, then we were thinking maybe about an imminent flu pandemic. But that prepared us so that in 2020, when the COVID pandemic arrived, we moved really fast. We initiated the Open Vaccine Challenge in March of 2020. And wow, a series of papers has been published from that initiative earlier this year, finally. And one of them, I mean, this almost seems like science fiction, but the company that brought much of the world COVID mRNA vaccines, Pfizer, they're co-authors in that paper, and they are testing Eterna designs called RLT10, which some of you know, Revalium designed that in a collaborative effort with many other Eterna players. And Bugakman's Lost, designed by Bugakman, of course. And so just, a couple of maybe three reflections there. That was, I think, is the first time in history that video game players and citizen scientists have been co-authors on a scientific publication with like a big pharma company. Second observation is that wouldn't have happened if it wasn't for Eternacon 5 in 2019. Third observation is that it also wouldn't have happened if we hadn't made a set of decisions over the previous Eternacons as a community, that we decided to make it so that the products that come out of Eterna, by products I mean the sequences, that we would make them available for open research by anybody in the world. That includes, of course, citizen scientists and open scientists, but we decided to go ahead and also make them usable by pharma companies, scientific research, for-profit entities. That was a difficult decision, but we made it. And that's what enabled this kind of unusual effort where the leading pharma company in the world is trying to see if the concept of superfolder mRNAs, again, developed on Eterna, whether those might work for actual mRNA vaccines, and they've de-risked it by testing the Eterna molecules. So that's really remarkable, and you're going to hear more about that from Hannah Wayman-Steele in the very next talk. Another thing that began over the last, let's say, 2 or 3 Eternacons is the idea of Eterna 3D, and we're going to hear more about that from CGEM, from Sarah and CGEM, as well as from Rachel Kresh tomorrow. But I'm just trying to think about what's going to happen in this Eternacon that might blossom in 2 or 3 years. And I have a suspicion it's going to be this Eterna 3D interface that we've started to pilot. I don't think we've yet found the amazing application yet. We're still in pilot mode, but boy, I strongly anticipate that in 2024, 2025, at those Eternacons, maybe that's going to dominate the scientific sessions of Eternacon. So that's speculation. We'll see. So the science that we do on Eterna is one of 2 just really amazing things that I personally value about Eternacon, that I think are personally very, very unique. The other aspect is it is community. It is trying to think about what do we want, how do we want science to happen, and what kind of community do you want to build around this really unusual mode of doing science. At every Eternacon, I think for the last 8 years, we've had panels, talks on the ethics of citizen science. And this year, we're fortunate to have David Resnick again later today to talk about the ethics of citizen science and lead a discussion. But starting about 2 Eternacons ago, 2020 and 2021, we realized that perhaps one of the most important ethical aspects of what we're doing is that we are increasing access to cutting edge science by communities that are actually kept away from doing science because of the expense and other kinds of structural barriers. And we started to put into place initiatives to ensure we could reach out to these underrepresented communities. And so I remember very well in 2020, right when everything was still shut down, hearing this wonderful talk from uh, the Prairie View A&M lab rats. If you remember, some of you were there. You remember that there were 6 incredible students from Prairie View A&M, historically Black college and university. They were planning to do a wet lab research with Dr. Gloria Regisford at Prairie View A&M. Everything was shut down, and we had reached out to them through Ronald Lorenzo, who's also one of the top Eterna players and also a professor there at Prairie View. And we had brought these 6 students to come and do the research instead virtually through Eterna. We had mentors like Astromon and Nando and others helped get them through the puzzle progression and help them contribute to that open vaccine challenge. And it was just an incredible presentation. And so that happened 2 years ago. And I was just checking in with Dr. Regisford about how those students are doing. Incredibly, all of them have continued in science, so they were able to surmount all the difficulties of that difficult period. They're still doing science or medicine. Some of them are doing master's programs, some of them are doing PhD programs. A student who was one of Dr. Regisford's lab reps in 2021 and also engaged with Eterna, Alexis Van Zandt, she was awarded a award where she became the HBCU STEM Queen and of 2022, or one of them. She was on the cover of Ebony magazine. She, along with a few other scientists, were actually the first non-celebrity people featured on the cover of the magazine. And in her interview there, she gave a shout out to Dr. Regisford and to the research that she did on RNA at CGEM and with Eterna. So that's just one example, and there's much more that's happening. You're going to see, for example, we're going to continue this year with a diversity panel led by Carmen Williams, who also has spoken in previous years about how representation matters in science and gaming, and Akira Bowler, who's the lead on our Twitch platform. And they brought in 3 pretty amazing folks from allied projects from outside Eterna to come tell us more about how to increase diversity and outreach to underrepresented communities. The other thing that's happened since 2021, this is just so in less than a year, is that we formalized a community engagement team. So prior to that, the developers on Eterna were focused on science and software engineering. So we decided, it's important to have a separate set of folks who are dedicated to building community engagement. And that community engagement team meets weekly, just like the software development team, and has, I think, an equal or larger number of people than the software engineering team. So think about that. So Eterna's developer community now is half of it is devoted to community engagement. And so I think some of you are seeing the effects of this, whether it's tweets and Facebook, whether it's outreach activities, the beginnings of potentially Eterna Academy. That was really initiated by Eternacon of 2021. Just a lot of us thought, who were developers, boy, we should just double down on community engagement. And we were finding that there were folks who were willing to come and work on that. And so you're gonna meet folks like Ana Mija and others who have joined the effort on that front. Okay, so to end, I kind of want to summarize what I think. Again, this is speculation, but what is next? I've already mentioned, I suspect 3D is going to be big. There's a lab going on right now called the Roll Your Own Student Challenge, and I want to challenge everyone who's at Eternacon, if you haven't submitted a solution to that lab, do it during Eternacon. It's in a very pilot stage where we think that the puzzles aren't hard to solve. We're just looking for people to make and submit complicated sequences that they think might form complicated folds, these so-called pseudonaut-like folds. And you can literally do it in a minute. I've done it like in a minute. And we're looking for as much diversity as possible in this set. Why? Well, let me again speculate about where this might lead to in 2 or 3 years. There's at least two major areas of science that could be impacted by what's beginning with the Roller Road Pseudonaut Challenge. So one area involves trying to accurately predict how given an RNA sequence, what does it fold up into in 3 dimensions? What is this complex fold? Some of you may have heard about an algorithm called AlphaFold that was released about a year ago that has transformed protein structure prediction in biology. It was trained on large data sets available for protein structure, which are not really available for RNA structure. But if on Eterna, if we could get examples of hundreds of thousands or millions or tens of millions of RNAs that folded into unusual folds, then that is getting to the point where you have a large, large data set. And when we have experimental data on those sequences, which seems feasible, that's a large data set that might enable an alpha fold for RNA. So can we get there? It would be really, really cool if in 2 or 3 years we're saying, hey, we made it happen. This is now the state of the art in predicting RNA structure, and it's all because of Eterna. And we start seeing, new shapes arise that are characterized in 3 dimensions using techniques microscopy techniques you'll hear about tomorrow. And then all of Eterna is looking at these 3 dimensional pictures designed by, by current players as well as players who've come in and been engaged over the next 2 or 3 years. The other area that I think the Roller Road Pseudonaut might impact is not just in being able to predict new structures or design new structures, but just to understand RNA structures that already exist in nature. So, I'll ask you this question. It's a little quiz. What do you think is the most studied RNA in biology right now? And just think for a second about that. So particularly those of you who are, biologists who are students in the field. Okay, E. coli, tRNAs, probably tRNA, but yeah, let's just tRNA. So it could be a particular tRNA, like a tRNA phenylalanine. I'm talking about a specific sequence. It could be an E. coli ribosome. I think that's what some of you are suggesting. In Wolf is saying microRNA or tRNAs. Okay, I would, if I limit it to one specific sequence, what do you think it would be? Could be the ribosome, like the E. coli ribosome. That's what Lorenzo's saying. All right. So I think those might all be the right answer, CRISPR. I think what Jill is right, though. The SARS-CoV-2 RNA genome, it's only been around for, I guess, 2.5 years. But boy, every biology lab in the world has taken a look at it and has done experiments with it. Every RNA biologist, for sure. So my guess is that might be it. But despite all that attention that's been put on that one RNA sequence, we don't know how much of it folds into interesting structures. We don't know if there are pieces of it that might form these pseudonaut-like complex folds. But if we knew that, A, it would be a new dimension for understanding how these viruses work biologically, and B, those structures could be great targets for new drugs that might help us finally take out this pandemic virus. And we also don't have that. So if we don't have that information for the SARS-CoV-2 virus, you can imagine we have close to zero information on pseudonauts or RNA structure in every other coronavirus, in flu viruses, in Bunya viruses, in Ebola and Marburg viruses. Some of you may have read that there's a Marburg virus outbreak that started two days ago. But what if Eterna could go and discover each of those interesting folded segments across all pandemic viruses so that we as a worldwide community can start to understand what they're doing and also start developing drugs that might attack them. So that's something and if you start expanding that purview to saying, we don't have an atlas of RNA structures in viruses, but we don't have an atlas of RNA structures in bacteria either, or in human beings. Our bodies are full of RNAs that haven't been characterized. That's basically a whole dimension of biology that hasn't been figured out. It's a whole kind of atlas. It's like discovering a new world and mapping out all the continents and the lakes and the mountains, and then giving them names, which could be Bugak Man's worm, right? Or it could be the very first design from Eterna was called Perception 2, I think. And so maybe Eterna players our community will be the one that maps out this entire RNA universe. And one way to do it is by going and trying to find pieces of natural RNAs that you think might have structure, putting them into roll your own pseudonaut, so that it's part of a big library of designs that will that Eterna folks and maybe others will start characterizing experimentally. All right, so just to conclude, so there's a lot that's going to be happening over the next 2 or 3 years. One thing that I think everyone has now noted is that RNA is a molecule that, you know, your siblings and your parents and your grandparents and your kids, like they've heard about what the molecule is. I've always believed that RNA is the most important like molecule on Earth, and it now looks like, you know, the rest of the world is agreeing. It may end up becoming the most important molecule in biology and medicine, at least. And I think the last thing I'd like to speculate on is, could Eterna be the medium by which all of us in this current generation and the next generation learn about RNA and become RNA scientists and RNA biotechnologists? Can we make it so that the Wikipedia entry on mRNA vaccines or CRISPR points to a beautiful tutorial, interactive tutorial on Eterna, and that's where folks learn about it? Could it be that by setting up the appropriate collection of puzzles and certifications on Eterna, could it be that everyone who goes into biology gets a certification on Eterna that they put on their LinkedIn or on their resumes? So that's the kind of possibility for what Eterna can do, and I think only Eterna can do over the next few years, that could transform not just science but also society. So I'm going to stop there. I went a little bit over time, but I'd like again to, okay, most importantly, to thank the organizers of Eternacon. LFP6 or Jonathan Romano, thank you so much for emceeing and setting up essentially every technical aspect that I'm aware of at Eternacon this year and last year. You've been at every Eternacon, I think, for the last 8 years, and it's, wow, it's so smooth now. We're really grateful to you. And I'd like to thank Amy Barish and Digital Embrace, Jill, for putting together the program of speakers and panelists. This looks like the best Eternacon ever. I feel like every Eternacon has been the best Eternacon ever because it just gets better and better. But I'm really grateful to both of you for putting this together. And lastly, I'd like to thank everyone who's continued to be part of the Eterna community, and those of you who are thinking of joining this community. Thanks for attending. I'm looking forward to lots and lots of great interactions over the conference. All right, thanks everybody.